Hello everyone, I'm Mariana and today we're going to talk about the result line of the program. The chosen subject was tolerogenic dendritic cell as an alternative for the treatment of experimental arthritis, and the group is formed by me, Leandro and Deusa. To contextualize our presentation, we are going to talk about arthritis firstly. Arthritis is among the most common autoimmune diseases in the world and affect around 1 to 2% of the population, besides being more prevalent in women. This condition is characterized by chronic inflammation of the joint that can lead to irreversible damage of loss of function, as you can see in the figure, where you observe a scheme of healthy joint and one with consequent arthritis injury. Arthritis is associated with a high risk of morbidity and premature death due to the early development of cardiovascular and interstitial lung disease. The mechanism to develop arthritis remains unknown.
However, researchers believe that involves a complex interplay between environmental risk related to unhealthy living habits or, for example, infectious disease and genetic factors. In this context, the onset of the disease probably involves the regulation of innate and adaptive immunity, triggering autoimmunity and different levels of inflammation and symptoms, but in a subclinical phase. Over time, the clinical phase of the disease is reached, being characterized by formation of autoantibody-mediated immunocomplexes produced by mature B cells, clonal expansion of self-reactive T cells stimulated by a variety of antigen-presenting cells known as APCs, mainly dendritic cells or DCs, and the spread of autoimmunity by formation of new epitopes and isotypes. Dendritic cells have an important role in immunity, linking innate and adaptive immune response through antigen presentation and cytokine production. DC can be found in the synovium of patients with arthritis, and they produce a large number of pro-inflammatory cytokines in addition to differentiation factors and chemokines which contribute to the propagation of joint inflammation. Immature dendritic cells are usually regarded as tolerogenic, whereas mature dendritic cells can exert either tolerogenic or immunogenic function depending on signals received during maturation from the microenvironment and invading pathogens. It is important to know the influence of those factors for the cultivation and obtaining of tolerogenic dendritic cells in the laboratory, and there already are some procedures in use, as you can see an example in the figure. Tolerogenic dendritic cells induce peripheral tolerance by induction of energy and deletion of T cells, blockage of T cells expansion, and induction of regulatory T cells or Tregs, being a promising possibility of arthritis treatment. That said, the hypothesis is that treatment with tolerogenic dendritic cells, known as TOL-DCs, improves the progression of arthritis, restoring immunological homeostasis in an experimental model. The general objective of the consulted works was to evaluate the effectiveness of treatment with TOL-DC in animal models of arthritis induction. We've gathered here the main specific objectives pursued by the lines of research that evaluate the action of tolerogenic dendritic cells in the treatment of arthritis. They are: induce tolerogenicity, evaluate immunomodulating profile of TOL-DCs, evaluate the effect of treatment with TOL-DC on the progression of arthritis, evaluate the effect of treatment with TOL-DC in vivo on the response of T cells. We are going to talk briefly about inducing tolerogenicity. Several strategies for generation of tall disease have been explored, including intervention with pharmacological agents, incubation with immunomodulatory cytokines, exposure to apoptotic cells, and genetic engineering through genetic deletion. In the studies consulted for the preparation of this work, the dendritic cells were obtained and differentiated from the bone marrow of mice. The maturation of these cells was accomplished by the addition of LPS or MLA in the cell's culture. Tau DC were obtained through intervention with pharmacological agents in the cultures, being EL10, TGF, dexamethasone, and vitamin D3 or genetic silencing. The results referring to the chosen works will always be represented in that order.
As we can see in these figures, in all works presented, diseases that receive different types of treatment, pharmacological agents and genetic engineers has low expression of MHC2, low expression of co-stimulatory molecules such as CD8, CD86, CD40. In addition, this disease showed high production of anti-inflammatory cytokines and low production of inflammatory cytokines. In physiological states, these are maintained in a mature state, presenting antigens to T cells in the absence of co-stimulation, and therefore maintain tolerance. During autoimmunity, disease maturated, which is followed by the upregulation of co-stimulatory molecules and activation of T cells that can discern self-antigens present by disease. Thus, co-stimulatory molecules play an important role in T cell DC interaction that could result in autoimmunity. Tau DC presents self-antigens to T cell in the absence of co-stimulation and can secrete immunosuppressive cytokines, leading to the inactivation of autoreactive T cells and the restoration of cell tolerance. Tau DC maturation is required under inflammatory conditions to inhibit autoimmunity. Thus, these results show that the different treatments were effective in inducing tolerogenicity in this cell. Once the tolerogenic profile of disease was confirmed, the next objective was to evaluate the immunomodulatory profile of these cells. For this, tolerogenic dendritic cells were co-cultured with lymphocyte, and the interaction between these two cells was evaluated. In all results, we can observe a decrease in lymphocyte proliferation, as well as an increase in the production of anti-inflammatory cytokines, including increased expression of FOXP3, which is a marker of Treg cells, and a decreased inflammatory response. This experiment demonstrated the regulatory effect of Tau disease on T cells. Thus, these results indicate that Tau disease are able to decrease the immune response in vitro. So, the next step was evaluate the effect of treatment with this Tau disease on the progression of arthritis in vivo. For this analysis, it is necessary to induce arthritis in animal model. The main model of arthritis inductions in mice are collagen induction models, collagen antibody induction, zimozan, antigen inductions, among others, in addition to genetic manipulation for spontaneous arthritis. It is important to note that all experimental models have particularities and limitations specific to the method. Most studies use collagen induction. After the arthritis condition was established in this model, the treatment with tau dendritic cells were performed. As shown in this figure, treatment with major tau disease was able to decrease the clinical and/or inflammatory score in different experimental modes of arthritis inductions when compared to control, resulting in a rapid decline in the severity of arthritis symptoms. The results showed that treatment with tau disease could reduce the pathologic score of joints. In order to investigate how this improvement in disease progression occurs, the response profile of arthritis animals treated with DC cells was evaluated. The next specific objective was evaluate the effect of tau DC treatment in vivo on T cell response. To evaluate the effect of treatment with tau DC in vivo, T cells were collected from the spleen of arthritis mice and different markers were used in order to elucidate the cell profile after treatment. According to this result, we can verify that arthritis animals treated with tau disease was an increase in the population size of Treg cells when compared to the control. And the treatment with tau disease was able to suppress Th17 cell populations and pro-inflammatory cytokines production. Together, these findings suggest that tau disease maintained their tolerogenic phenotype and function in vivo, thus promoting a potent tolerogenic microenvironment leading to dramatic reduction in arthritis symptoms and amelioration of disease progress by modulation T cells functions, upregulation Treg cells, resulting in the upregulation of anti-inflammatory cytokines and the downregulation of pro-inflammatory cytokines. In conclusion, tau disease could restore immune tolerance through the induction of Tregs, as well as the suppression of DC-T cell interaction. Moreover, tau disease represents a potentially promising immunotherapeutic strategy to manage arthritis and other autoimmune diseases. Bibliographic references. Thank you so much for your time.